Using immunoprecipitation to isolate a protein. The first step is to break open the cell membrane, releasing the cell contents. This can be done mechanically, using chemicals or sound waves. Once the cell membrane is broken up, the hydrophobic phospholipids will associate together, forming small liposomes. The aqueous proteins have been released and are in solution. The goal of immunoprecipitation is to isolate a particular target protein. Some information about the target protein must be known so that antibodies against the protein can be generated. The primary antibody is a Y-shaped protein. The forked ends of the Y are variable regions that are specific to a particular protein, in this case, the target protein. In order to detect the binding between the primary antibody and the target protein, a second type of antibody is used. The secondary antibody has been specially modified to have an added magnetic bead. This will allow for separation of the secondary antibody and anything it is bound to. The secondary antibody is specific to the primary antibody and will bind to any primary antibody present in the sample on the constant region, or stem part of the Y. The binding between the antibodies and the proteins is happening in the aqueous solution and is not visible to the naked eye. In order to detect the secondary antibody modified with a magnetic bead, a magnet is held outside of the solution. The magnetic bead is attracted to the magnet, so the secondary antibody is anchored to the magnet on one end and bound to the primary antibody on the other. The primary antibody is bound to the target protein. That is how immunoprecipitation can be used to isolate a target protein using the specificity of antibody binding.